Good evening. Today we will be going over lesson 9.3. Before we start the main points, we're gonna go over two vocabulary words. Apoptosis, controlled cell destruction. Cell suicide, it's where the cell kills itself. Stem cells, embryonic stem cells, found in developing fetuses or embryos. Adult stem cells, found in adults, and their job is to repair damaged organs. Now, both of these cells can turn into any kind of cell, but for different reasons. Embryonic stem cells build up the body from nothing. Adult stem cells repair the body. Onto cyclins. What are cyclins? Cyclins are regulatory proteins. Their job is to start different stages of the cell cycle or start different processes within the cell. Different combinations of them will do different activities. So let's say you put cyclin A with cyclin B, gives you process C. Cyclin A with cyclin Y gives you process X. So the same cyclin at different amounts with a different kind of activation lock will give you different things. One of the processes that the cyclins are responsible for is protein synthesis. Another one, DNA replication, nuclear division, the list goes on. Now, what do they activate? They activate the cell cycle, but the cell cycle itself, can it simply just activate? Can the cell do what it wants at any time? No. Because for the cell cycle to go on and on, there are certain checkpoints, certain conditions to be met. Example given here is the G1 checkpoint. The cell here is gonna check for two things in particular. Is the cell big enough to start the synthesis phase and prepare to divide? Is the environment suitable? Do we have enough space for two daughter cells? Do you have enough space to even do the division? So on and so forth. There is the G1 checkpoint. That checkpoint happens before synthesis, and the checkpoint happens before G2. Now, what happens when this whole system functions correctly? You get a living cell. What happens when it doesn't function correctly? You get three cases. You either get a cancer cell, you have to undergo apoptosis, death, or the cell functions as it's meant to be for a bit, which we'll go over later. Now, example given here is a cell's DNA is damaged by the environment. Let's say exposure to radiation from the sun. The DNA damage has one of two ways. Successful repair, none of this happens, normal cell, or failed repair. This failed repair will lead to these three side effects. The cell might not be able to undergo apoptosis. It cannot self-destruct even though it's damaged. And it is unable to, let's say, contain the part of the DNA that's damaged. It's unable to stop it. No tumor suppressor genes. And when both of those happen, then the cell might stay there, and then another mutation happens, and it happens to activate the growth oncogenes. And you just notice that the cell is just reproducing without an end. Now, you may think, well, more cells, that's useful. But at the same time, the lack of organization might lead to abnormal cytoplasmic structure. The cell can no longer do its job. So the cell keeps on reproducing, skipping a lot of time and interface is skipped, and taking a lot of space, ignoring the checkpoints, and you get a tumor. Now, we were talking about carcinogens, but we didn't go in depth. A few examples of carcinogens you can look onto here is tobacco. Smoke is known to cause lung cancer. Sunlight, air pollution, and a very interesting one is virus infection. It's very important to be vaccinated against viruses because viruses can alter DNA structure. And altered DNA structure, as you'll see soon, can cause, let's say, future cancer rather than cancer right now. So let's look at a flowchart here. We have a normal cell. The normal cell incurs cell damage. Either gets a mutation or fixes it. Let's say it gets a mutation. This is mutation number 1. Nothing happens. Cell functions just normal. More cell damage. Mutation number 2. Nothing happens. Cell functions just normally. Third mutation happens. Cell still functions normally. No worries. And then at a fourth mutation, the cell can no longer handle it. The cell can no longer just function. It causes something to happen, and then you get a malignant tumor. Now, you may think, okay, that happens within the cells, but that same thing is the same reason cancer can be inherited, God forbid. Let's say this person A, has cancer. He has a prostate cancer. He gets it treated, he lives on, he has 2 daughters, and so on and so forth. He still has cancer-prone genes. One daughter lives her whole life fine, has 2 kids, and no issues whatsoever. The other daughter ends up with ovarian cancer because the hand she was dealt, or the genome she was dealt, was inherently damaged. It was more prone to cancer. It had 2 mutations, for example. It only needed to be damaged 2 more times rather than 4 times. So she gets ovarian cancer, but her sister is fine, has kids. The sister here has a son, and the son doesn't have cancer. He's still far too young, but he's considered at risk of cancer because she has cancer genetics that would lead to prostate cancer, for example, but she hasn't had it. So she's fine, but her son is at risk of cancer because his cancer genetics are more likely to function in him than they did in his aunt. And that is how cancer is inherited. You get it, let's say, from a grandparent. Your parents are fine, but you're at risk of cancer. And that is all for lesson 9.3. Enjoy your vacation.